fall allergies. Good afternoon. I want to tell you just a few things about Dr. Sands. Dr. Jeff M. Sands is the Juha P. Kokko Professor of Medicine and Physiology and Director of the Renal Division at Emory University School of Medicine. He's a graduate of Harvard College and Boston University School of Medicine, as well as the University of Chicago and National Institute of Health's Lung and Blood Institute. Following fellowship training in the laboratory of kidney and electrolyte metabolism at the NIH, he proceeded to a clinical nephrology fellowship at Emory University. Joining the Emory faculty as an assistant professor in 1989, Dr. Sands was promoted to associate and then full professor. He accepted the Kokko Professorship, and division director position at Emory in 2002. He served as executive vice chair of medicine from 2009 to 2015 and as associate dean for clinical and translational research from 2006 to 2010. He currently serves as president of the American Physiological Society and is principal investigator of several key renal research grants. Dr. Sands is a co-founder of Nephro DI Therapeutics and is considered a key opinion leader for nephrogenic diabetes insipidus by the National Organization of Rare Diseases. And we welcome Dr. Sands today. Thank you. So you've got a mic. So.
I've got a mic.
Yes.
Thank you, Dean Lathan, for that very kind introduction. I hope, can everybody hear me? Okay. It's a pleasure to be here today. And let's, if I can, sadly, I got to put these on just to see the little icon. I got to click here. So here it is. Okay. So what I'd like to tell you about today is some work that we've done. Russ Reed saw some of this earlier this year at the CED conference in North Carolina and was kind enough to invite me to come today, talking about physiologic insights for novel therapies of a rare disease called nephrogenic diabetes insipidus. And as you can tell from the screen, this is a disease that affects children. Okay. So disclosures, as Dean Latham said, I'm a co-founder of a small company, Nephro DI Therapeutics. This is a startup that was started in August of 2017, and I'm a consultant for Third Kidney Incorporated. The latter has nothing to do with anything I'll talk about today. So I have some learning objectives. I wasn't sure if we needed learning objectives, but I figured if there were students, you're supposed to have learning objectives. So what we'll do is we'll review nephrogenic diabetes insipidus, review the physiology of the urine concentrating mechanism, review regulation of water transport by vasopressin, and discuss non-vasopressin pathways to increase urine concentration. And it's the last bullet point that really forms the basis of the novel therapies and what we hope will someday be a treatment brought forward by our startup company. So you can see this poor boy here obviously is holding it in. So what is nephrogenic diabetes insipidus? It is the inability to concentrate urine. I suspect no one in the room has ever heard of this disease because it's a fairly rare disease. It has absolutely nothing to do with diabetes mellitus. In fact, it's actually unfortunate that diabetes mellitus and diabetes insipidus share the word diabetes, because every time we send in a patent request, the patent officer thinks we're talking diabetes mellitus and sends back a whole bunch of questions that we then say, no, you've got the wrong disease. But both of them result in extra urine production. In the case of nephrogenic DI, these kids can make up to 20 liters of urine a day. I think that's a liter an hour, 24/7, 365. That's an enormous amount of urine. For the young babies, think about how many diapers the poor parents are changing every day, and you have to give the child that much water or else they get dehydrated. There are two forms of congenital NDI, one of them related to two different mutations. Let's see, is this the pointer? No, what did I hit here? Okay, I hit the wrong button. I need, how do I undo that? Escape. Hmm? Okay. Oh, I got rid of it. Okay. Yep. I guess that wasn't to point. I'll use the mouse to point. There are two forms. One form is related to a mutation in what's called the AVPR2 gene. This is the type 2 vasopressin receptor gene. I'll show you more about that later. This forms an X-linked pattern of inheritance, meaning that mothers are carriers. They have one mutant X chromosome and one normal X chromosome, but they don't have the disease. And then if their male offspring gets the wrong X chromosome, the boy ends up with the disease. So this form is only occurs in boys. It's 90% of the cases of congenital NDI. There are rarer forms that are due to a mutation in what's called AQP2, or the Aquaporin 2 water channel, the actual protein through which water gets reabsorbed. This is much rarer. It's mostly autosomal dominant, so either boys or girls can get this disease. It's about 10% of cases. As you'll see as we go through the talk, the thinking on my part was that if the receptor was mutant, well, everything else downstream was just fine. Can we find a way to bypass the mutant receptor and activate the rest of the machinery so these boys can reabsorb water? Unfortunately, for those who have Aquaporin 2 mutations, that strategy won't work because it's the actual water channel itself that's mutant. And until we get to the days where CRISPR-Cas is able to do gene editing, I'm not sure there would be a therapy for those, but that's the much rarer form. Their current treatments are ineffective. What do we do? Basically, these poor kids have to drink 20 liters a day to match how much urine they're making. And the other therapies are minimally effective, to say at best. And if we could reduce the urine output by, say, even 20%, that would be clinically significant for these kids. It would make their lives much better. Certainly, if we could get it down from 20 liters, let's say, to 10 liters, a 50% improvement, it's still an enormous amount, but it would really improve the quality of life and decrease the chance that they'll get complications from the polyuria. So, in terms of the disease, you can see this young boy here has a very large sippy cup in his hands, and he's drinking lots of water. Congenital NDI is considered an orphan disease. That gives you certain, when you're thinking in terms of a startup and potential entrepreneurial aspects, an orphan disease has certain advantages for getting through the FDA. In particular, a pediatric orphan disease has other advantages. How rare is it? It's roughly 4 to 8 boys per million births will have this disease. So this is really not common. 4 to 8 per million boys. And what happens as a consequence of the disease? Well, if you don't keep up with your water input, if you don't drink all that water, you can have severe complications. You can get seizures, even coma from the complications of dehydration. You can get permanent brain damage. In the literature of the 1980s, it describes the genetic syndrome as including mental retardation. But in fact, it is not part of the syndrome. In the early 90s, when these genes that I mentioned were cloned, at-risk families were screened, the baby boys were treated with water. And they don't develop mental retardation. I've met college graduates with this disease. It was a completely preventable form. Now, why it required knowledge of the genes to treat at-risk kids with water, I don't understand, but that's the history of it. You can get developmental delays from dehydration and the cognitive impairment. So how do you prevent these complications? Well, you have to worry about the dehydration. Another aspect is frequent urination. These kids need to urinate 20 liters a day, and they tend to hold it in. They just don't want to be running to the restroom every hour on the hour, so they tend to hold it in. Their bladders begin to get big, and then they can get consequences of urinary obstruction just because they're holding it in. And the babies can get failure to thrive. Fortunately, these days, in families where you know there's a history, there's a good recognition. But in families where this is the first presentation, sometimes there are delays in bringing these kids to medical attention. So hydration is a key. You need constant access to water and constant access to restrooms. Many of the babies need either a G or a J tube to get enough water in. For anyone who has either fed a baby or has fed other people's babies, you know babies tend to regurgitate a fair amount of what they take in. Well, that's with normal feeding. Trying to get a liter an hour into a baby's stomach is a real challenge. So particularly in Europe, the standard is to put a tube into the jejunum. It bypasses the stomach and puts the water directly in, and these kids will need it till they're about age 2 to maybe 3. In the US, this is less common, mostly because insurance companies don't want to pay for it until you prove that there's no other way to do it, and then usually they might need it as well. You need to give them an extremely low sodium diet, 0.5 grams a day. In heart failure, we try to get patients to eat 2 grams of sodium per day. And that's very hard to do, and most of our patients, frankly, don't do it. But that's the goal. This is a quarter of that. This is kids going to a birthday party and standing over McDonald's French fries and just inhaling. I don't know if they even have to eat the French fry, but it's really terribly restrictive. And so in addition to all the water they have to drink, it really is a problem for them. I mentioned that there is minimally effective therapy. You can give diuretics. Now, it's going to sound counterintuitive to give diuretics to somebody who's making 20 liters a day, and the mechanism by which this works is somewhat complicated, but giving thiazide diuretics does have a small benefit. You need to give potassium supplements to make up for the thiazide, and you can give nonsteroidal anti-inflammatories like ibuprofen, naproxen, any others, but these are all nephrotoxic, toxic to the kidney, so we really hate to give a nephrotoxin to somebody who may have a kidney issue. And even with the standard of care, complications are frequent and the quality of life is poor. You can see at the bottom, just as a schematic, how many bottles of water these poor kids are drinking. I mentioned the normal complications are in the bladder, particularly urinary tract distension. Taken to an extreme, they can get urinary tract rupture, infection, pain, improper bladder function, and if it's really left untreated, they actually can end up with renal failure from chronic obstruction. And so the kids, and as they progress to be adults, get a renal ultrasound every year to make sure that they're not inducing chronic obstruction leading to kidney damage. So, as I mentioned, they drink 20 liters a day, and they urinate 20 liters a day, and they have to drink an equal volume. Current therapies minimally effective. So a novel treatment for congenital NDI would meet the criteria for an FDA Orphan Drug Act status. It's likely to qualify for an FDA Rare Pediatric Disease Priority Review voucher. And what that is if the FDA, to encourage people to develop drugs for rare diseases, especially rare diseases in children, offers a voucher which can then be used for an expedited review of any other product through the FDA. So as you can imagine, a disease that affects 4 to 8 per million is not going to have a big market. I mean, no company's ever going to be able to make money selling, no matter what you charge, selling something to that smaller group of individuals. But these vouchers are very valuable, I'm told. We don't have one. You have to bring the product to market to qualify, but then those can be used for other purposes. So let's say a company gets a voucher and then they either sell it or they're acquired by another company and they have the next Lipitor. And they can bring the next Lipitor or multi-billion-dollar drug to market 6 months faster. You can imagine that for that product, these vouchers are extremely lucrative. So that's one of, in terms of the entrepreneurial business aspects, the potential for getting a voucher is very important. And we've developed a lead compound called NDI 5033, and I'll show you some data suggesting that this may be a potential new therapy. So, given all that, I thought I would go through how the kidney actually concentrates urine to provide the biologic basis for what our therapy will hopefully do. So here's a kidney. You can see this is in cross-section. So here's the kidney is composed of a cortex. It has the ureter at the other end. The medullary pyramids go down from the cortex into the renal pelvis, and there are some calyces. So this is a very schematic view of the kidney. It's the medulla, this portion here, that is critical for concentrating or diluting the urine. And I'm going to walk you through what is some fairly complicated physiology, but try to get at the basics of how this all works. So this is how I take the previous slide and I simplify it to this. So this is the glomerulus, this is the loop proximal tubule, the loop of Henle, the distal tubule, and the collecting duct going through these zones. Now, up here in the proximal tubule, there is a lot of salt and water reabsorbed. It's done isosmotically. There is no separation of salt and water in this part of the kidney. So for purposes of concentrating or diluting your urine, this segment is irrelevant. Now, I know many of my friends up the road at Wake Forest who live and die with the glomerulus would be horrified to see me simplifying the glomerulus this way. I don't mean it's unimportant, it's just not important for this particular topic. The action, oops, what did I hit here? The action comes here in what is called the thick ascending limb of the loop of Henle. There is a transporter here that moves sodium, potassium, and chloride from the lumen of the thick ascending limb into the interstitium. The potassium recycles futilely, so the net result is you take salt out of this limb, of the lumen of the thick ascending limb, and add it to the interstitium. You are separating salt and water. This is the transporter that, if any of you know patients who take Lasix, for example, this is what's inhibited by Lasix. It's a powerful loop diuretic, and what it does is block this transporter. But if you're not on Lasix, for normal individuals, by taking salt out of the lumen here, you deliver very dilute fluid up here, as dilute as 50 milliosmoles. And in the case where you need to get rid of a lot of water, like let's say you just had 4 cups of coffee to get ready for your morning lecture, well, you're going to make very dilute urine. And this dilute fluid can simply be excreted unchanged through the collecting duct down into your bladder. But if it was earlier, let's say when you first got up this morning, and if you weren't pulling an all-nighter, then you weren't drinking any fluid overnight, you need to make concentrated urine. The salt that's been added here to the interstitium makes a hypertonic environment. Okay. And due to the presence of water channels, these aquaporins, in the apical membrane of the collecting duct, water can be reabsorbed under the action of vasopressin or antidiuretic hormone, and with that hormone present, with a hypertonic interstitium, and with functional water channels, you can reabsorb water and produce very concentrated urine, and that's what most people are doing first thing in the morning. Now, as that water is taken out of the collecting duct, the solute that's left behind, the urea and sodium chloride, are progressively concentrated, and then the urea can be reabsorbed here in the deepest inner medulla to further concentrate the urine. So this is the basics of it, and the key takeaways are that you need a functional transporter here to separate the salt from the water, to deliver dilute fluid here, which in the absence of vasopressin, you simply excrete. When you have too much water in your body, you can get rid of it. And if you need to concentrate, you need a functional receptor, you need the hormone, and you need the water channels. So there are many water channels that have been cloned. There are now 13. I've focused up till now on the ones in the collecting duct, in particular Aquaporin 2. There are others. A Nobel Prize in Chemistry was actually awarded in 2003 to Peter Agre, who, from Hopkins, who did the original cloning of the first Aquaporin, which was Aquaporin 1. Just so that you can see, there are many there, but for the purposes of today, the one in green is the focus of the talk. And here's what it looks like. I think this is interesting. It doesn't have anything to do with the product we're developing, but the way Aquaporin 2 actually exists in the membrane is it's a 6-transmembrane spanning region, and then it folds up to this shape here, and the water actually goes through this pore here. And in a biological membrane, there's actually 4 of them that sit in the membrane, and water can go through each of these cylinders. So how does we take this, and let's think about how we can apply this to the question in hand. So this is the normal biology. So here you've got a cell. This is the urine-facing side of that collecting duct cell, or the lumen. This is where the fluid will eventually become urine. This side here is the blood-facing side. You can see that vasopressin, abbreviated AVP, binds to its receptor, the V2 receptor. It activates signaling molecules that eventually activate a kinase called protein kinase A. Protein kinase A then phosphorylates the Aquaporin 2 molecule, moves it to the apical membrane, and water can be reabsorbed. I've added here a molecule we've not talked about, another kinase called AMP kinase. AMP kinase is best known as a metabolic kinase. It's involved in cellular energy metabolism, but it is there. And normally, in normal physiology, it actually doesn't do anything. It's just kind of there. We've looked by Western blot, we know the protein's there. It's actually repressed when pKa is present, so it's shut off. I think of it as that little runty tire that's in the trunk of your car that you hope never to put on your vehicle. It's your spare tire. It just kind of sits there hoping you never need it. But, as I'll show you, we think you can make use of it therapeutically. This is just to show you what the V2 receptor looks like. The red circles are all the amino acids that a family has been described with a mutation at that location. This presents problems for diagnosis because, and this is from 13 years ago, there would be more red circles on a newer picture. So it's not a monogenetic disease. It's not like sickle cell anemia, where every patient with sickle cell has the same mutation. In this case, every family, more or less, has its own mutation. So genetic testing is a challenge. You have to know what the mutation is that runs in the family if you want to screen at-risk baby boys to see if they have the same disease. Now, they do get screened, but if you know there's a history, it doesn't take long to figure out that the baby boy is affected or not affected. But doing the genetics is done. There's an investigator in Montreal, actually, who will do this for free, basically. As long as you FedEx the blood to him, he'll run the genetics. Aquaporin 2 mutations, and this is what it looks like, are much less common. Again, you can't really screen for it easily because it's a whole bunch of different mutations. So what happens in disease, in the disease of X-linked NDI? Well, again, here's the picture of the normal situation. I've gone through this before. Let's look at the disease. So if you have a mutation in the V2 receptor, vasopressin can't bind to it, the receptor is not activated, all of these in-between signaling molecules are not activated, and pKa is not activated. But the repression of AMP kinase is relieved. And we reasoned that since the aquaporin 2 molecule is just fine, if activation of AMP kinase could now do the job that pKa normally does, perhaps that would be a way to help these kids make less urine. Okay. So here's the proposed solution. X-linked congenital NDI is due to mutations in this V2 receptor. The aquaporin 2 water channel is normal, but it cannot be activated by vasopressin because the receptor is mutant. And so we need to find a non-vasopressin sensitive pathway to activate aquaporin 2. And our target, our therapeutic target, is AMP kinase. The long name is 5' activated protein kinase. I mentioned that it plays a role in cellular energy balance. And I was read a review article. This is actually true. I was actually at listening to a seminar, as you are today, and the speaker was talking about AMP kinase and happened to mention that it can stimulate NKCC2, that transporter in the thick limb that's inhibited by Lasix, and it can do it independent of vasopressin. And I thought, aha, maybe if it also would have an effect on aquaporin 2, that it could do the job that vasopressin normally does. In addition, osmotic stress stimulates AMP kinase, and the place in the body with the most osmotic stress is in this part of the kidney. So after the seminar, I went back to my lab and asked members of my lab, has anybody ever looked at whether AMP kinase does anything in the inner medulla or have an effect on aquaporin 2? And you just never know when dots are going to get connected that just haven't been connected before. And as you can imagine, since I'm here, those dots turned out to be worth connecting, or I wouldn't be talking about it. So how did we go about establishing the biologic basis that maybe AMP kinase should really be a therapeutic target? The first thing we did was in the test tube. We took a construct that had either aquaporin 2 or UTA1 and incubated it with commercially available AMP kinase, so we bought from Sigma, and just mix them in the test tube and look to see if the phosphorylation of aquaporin 2 or UTA1, the urea transporter, was increased. We knew from work that I didn't have time to show you that when you phosphorylate aquaporin 2 or you phosphorylate UTA1, you activate the molecule and you move them to the membrane where they need to do their job. And you can see, I think even from the back of the room, that when you add AMP kinase, the phosphorylation of these two key proteins goes way up. So we said, great, this shows you can directly phosphorylate these proteins, at least in the test tube, with artificial AMP kinase. Excuse me. The next question was, could we do it in actual kidney tubules? So my colleague Janet Klein, who's also a co-founder of Nephro DI, did these studies. She took rat intermedullary collecting ducts, so she takes the rat kidney and she makes a suspension of intermedullary collecting ducts, metabolically labels with P32, and immunoprecipitates. And we used a drug called metformin to activate AMP kinase, because for the tubule, we couldn't use the artificial AMP kinase, but we used a drug that does activate AMP kinase. Metformin is used widely to treat diabetes mellitus, which also doesn't help us with the patent reviewers, because now we're confusing them by using a diabetes mellitus drug for diabetes insipidus. But metformin does it. And you can see most dramatically for aquaporin 2, 2.5-fold increase in the phosphorylation of aquaporin 2 in actual kidney tubules in the presence of metformin. UTA1 also goes up significantly, although the magnitude is smaller. Well, that's nice, but does it change function? And so we did this study where we took these kidney tubules and we perfused them in vitro. I'll explain this to you a little bit. So what you have to do is microdissect out a kidney tubule. These things are, on a good day, you can get a 0.5 millimeter long tubule, and it'll be 20 to 30 micrometers in diameter. So you take this little tiny straw-like structure and you mount it on pipettes. And you put one pipette into the lumen from one end, you can put fluid in, you collect the fluid out the other end. It's really a technical tour de force, but it's really a blast for anyone who's done it to actually be able to pull this off. Luckily, I have younger people in my lab who still have the hand-eye coordination that I used to have, but I don't have anymore. And what you can see is water permeability goes up after 30 minutes of metformin, and this is a marked increase. Same is true for urea transport. We now used another compound, ACAR, that also activates AMP kinase. Both did it. Okay, so we can phosphorylate it. We can show an increase in transport. What about the real key, urine osmolality? So we took a mouse that was made at the NIH. That's a V2 receptor knockout mouse. And this mouse mimics beautifully the human disease of X-linked NDI. And what we did is put the mouse in a metabolic cage and either measured urine osmolality, either without metformin or with metformin, and you can see within 5 hours urine osmolality has gone from a very dilute urine. Anything under 300 is considered dilute. So the controls stay under 300. The metformin-treated animals go up very nicely. And in fact, the magnitude of the increase is much more than what you typically call a positive response in people, where if it goes up by 200, you're very happy in the clinical world. Well, this is only for 5 hours. We're talking about a genetic condition where these kids would have to be treated for life. So 5 hours is not going to cut it. So we did this protocol here. And what we did is, unfortunately, the mice are rather fragile, and so they don't live all that long. So we took rats, plus they're bigger and make more urine, easier to study. And we took rats and gave them a drug called tolvaptan. And tolvaptan inhibits the V2 receptor. So although this is not a genetic knockout, it's in essence a chemical or pharmacological inhibition. And this drug is actually used to treat patients as well, but not with the goal of inducing NDI, although it does seem to do that. And so you take the rats, you get basal urine. On day 2, almost all the rats get tolvaptan, and on day 3, half of them also get metformin, the other half still stay on tolvaptan only, and on day 6 they're sacrificed. I won't show you, but we did a control group to look at metformin alone, which really does nothing in the control animals. And here are the results. So rats make a urine of around 2,000 milliosmoles. With 1 day of tolvaptan, it's dropped almost in half. The open bars stay only on tolvaptan, and they stay at the same level, whereas the striped bars get the metformin, and you can see it goes up within a day, more at day 4, and it's still up at day 5. So this effect, at least up in this point, did not show tachyphylaxis. It seems to be a nice sustained response, in fact, back to control levels. We went further and looked for changes in the proteins. This is the urea transporter UTA1. You can see it goes up 60% at the end of the study. Aquaporin 2 goes up by 44%. We looked at another form of Aquaporin 2, a specific site that was phosphorylated at serine 256. This site is associated with moving Aquaporin 2 into the membrane. So it would be nice if this went up, and it doubles. And another site, 261, is associated with pulling it back out of the membrane. This would decrease water transport, and it doesn't change. So you've added more aquaporin 2 to the membrane by phosphorylating serine 256, and you do not activate the retrieval mechanism serine 261. Well, can we carry it out further? And we did. We said, well, okay, 3 days is nice, but again, we're talking lifetime therapy. So we repeated the same experiment, but now we went out longer. In this case, we really wanted to block the animals with the tolvaptan, so you can see that they get tolvaptan now for 3 days before we start to intervene with metformin. And in the green or turquoise are tolvaptan only. They stay at around 1,000. The ones that get metformin in purple begin to go up at 1 day, and a statistically significant increase back to control levels that was sustained out to day 14 of the study, or almost 10 days of therapy. So we thought, and the animals seemed to do just fine. Well, you might say, well, why don't we just give metformin to people? It's commercially available, it's dirt cheap. You can go to the drugstore with a prescription. At least in Atlanta, you go to the supermarket and you get it for free. I mean, you go to the local Publix. I don't know if you have Publix here. It's free. They basically want you to, while they're taking their time counting the pills, they assume you'll do your food shopping, and they'll more than recover the cost of this dirt cheap drug. The problem is, what I haven't told you is that this dose of metformin, if you were to equate it to a person, you just can't give them that much. They get too much GI distress. It just, and in fact, we tested a few kids in Atlanta, college-age kids, because we could only do it for 18 and up. And there was an intriguing transient result, but not one we could hang our hats on. And we knew we'd have to dose escalate to the point that it wouldn't be tolerable. So we went back to our model where aquaporin, you can see here, you've seen this picture before now, vasopressin binding to the receptor, activating pKa. This is normal, inhibiting AMP kinase, and water gets reabsorbed. In NDI, if you have a receptor mutation, or in our model where you give tolvaptan to block the receptor, no pKa activation, AMP kinase does its work, and metformin can mimic that or activate AMP kinase. So, oops, yeah, I said that. So we came up, about this point in time, I met a pair of medicinal chemists in Georgia. A mutual friend, a former Emory faculty member, actually read my papers. He said, I have some friends you got to talk to. These were medicinal chemists who had other AMP kinase activators, which they were developing as a treatment for diabetes mellitus. And pharma has put a lot of money into AMP kinase activators for the treatment of diabetes mellitus. Well, these didn't work, but it was perfect. We said, that's great. We don't care that they don't lower glucose. Can they work to activate AMP kinase in the medulla and improve water handling? So we tested 3 compounds, and one of them, NDI 5033, turned out to do everything that metformin did, except it did it better. It did it at a much lower dose with more potency that one would predict, if the same dose could be used, is taken to people, would be tolerable. And most importantly, it had no effect on blood glucose. So here's the data. There were 3 compounds. You can see NDI 5033 almost brought the animals back to basal levels. This is in the tolvaptan-treated rat model. These other 2 compounds had some benefit. This one a little bit statistically, but certainly not as good as the 5033. And there are differences in the molecules themselves that may be responsible for why the 5033 worked and the others didn't. Of course, other members of the family were developed to treat a different disease. We then went and did dose response. You can see here we went up progressively, 5, 10, 25, 50. It worked. We also did it in the V2R knockout mouse, we wanted to be sure that the AMP kinase activator, whether it be the 5033 or for that matter metformin, wasn't simply knocking tolvaptan off the receptor. And the reason you had a benefit was not because it was doing what we thought it was doing, but because it was preventing inhibition of the V2R. Well, the V2R knockout mouse is genetically knocked out, and so showing the same effect in that animal model was convincing that this really was through the mechanism we were suggesting. Now here was what we were encouraged to do as the quote, hate to use the term, but they call it the killer experiment. And one of the differences between doing NIH science and doing science for drug development, entrepreneurship, to interest venture capital investment, we hope, is NIH, you do this step, then this step, then this step, and you dot every I, cross the T, look at the mechanisms. Here they said, no, go to the end result. Do the killer experiment, and you can go back and fill in the blanks later. It's a totally different way of thinking about things. I see Russ shaking his head, so I think he probably agrees with that. And what we did is we took rats, used the tolvaptan-treated rat model, and we said, what would real live kids do? Well, they go to school, and then after school they go out and they play. And and they're going to run around, and they're probably not going to eat real well because they're too busy playing to want to be sitting there and eating. So if we're giving an AMP kinase activator, we have to make sure that when kids are active, they're not in some way getting hypoglycemia, because then we'd be hurting the kids. So obviously we couldn't do this in kids, so we took rats and we treated them. You can see in the top panel here, this was a longer study. Here half of them get tolvaptan only, the others get treated with the NDI 5033. And on these 5 days here, what we did is we took their food away in the morning, and then later in the day, we put them on a rat treadmill. They make treadmills, and you can get the rats to run. And we exercise them for 30 minutes. In fact, the technician in my lab who does this also coaches girls volleyball at the high school level. So she knew how to motivate people, at least kids, how to exercise, and she made sure that these rats ran. And what you can see here is that whether it was before exercise in the light blue or after exercise in the darker color, the red lines here show you the normal range of blood glucose. They were within the normal range the entire time, either before exercise but still being fasted, or after exercise with both. So they never get hypoglycemic. They never leave the normal range, and urine osmolality goes up. So how does all this fit together? Again, you've seen this now, water through Aquaporin 2, vasopressin binding to its receptor, activating adenylyl cyclase, generating cyclic AMP, activating pKa to phosphorylate Aquaporin 2 and move it into the membrane so water can be reabsorbed. AMP kinase just sits there. In NDI, whether it's due to a V2 receptor mutation, tolvaptan binding to that receptor, you get no pKa activation of AMP kinase through NDI 5033 to phosphorylate Aquaporin 2 and move it to the membrane. So we think this is a potential therapeutic strategy where we've basically bypassed the receptor mutation, and found another way to activate the molecule that moves the water. Now, this is, as what I've described now, has all been congenital NDI, a rare disease. But there is a condition in adult medicine that is much more common. When patients are given lithium to treat manic depressive disorder, and lithium is still the best treatment for manic depressive disorder, they develop an NDI. Not all the patients, but about half the patients will, and if they do, once they get it, it's very hard to reverse. Now, how does lithium do that? Well, lithium works, here's lithium, and it inhibits the adenylyl cyclase to cyclic AMP generation. So the result is that you get no cyclic AMP, you do not activate pKa. So it's slightly downstream of the genetic mutation, but again, these middle molecules are blocked by the lithium, pKa is not activated, and we think that NDI-5033 could activate AMP kinase in the lithium-treated model. Now, this is theoretical, as we have not done these experiments yet, but there are probably 2 to 4 million Americans on lithium, and if this worked, lithium could be more given to more patients, because lithium is also dirt cheap, although who knows what NDI-5033 would cost, but it's the best therapy. So this could keep people on lithium, and patients who are well controlled on lithium in terms of manic depression, when you try to take them off because they're developing NDI, they don't want to come off. It's because their mental illness is controlled and they're very reluctant. I've been called by psychiatrists who say, I have to take my patient off, but they won't do it. They'd rather have NDI than have more flares of their manic depression. So this would be a way potentially to help those individuals. So here's our development plan. Potentially this is a very fast pathway for drug development. I won't go through all the details here, except to say we're doing many of the pharmacology and toxicology studies at present. These other steps are coming. We're going to need of course, more money to do many of the later steps. But we have the money through the Coulter Research Foundation and the Georgia Research Alliance to do much of the initial pharmacology and toxicology. But the reason it could make it on a time frame of roughly 4 years and a total budget of only $15 million. I mean, $15 million to me is a fortune, but in terms of drug development, $15 million is like nothing, is that these compounds already exist. Much of the initial tox and PK have been done. Some of the related molecules have even made it into phase 1 trial, so we think that bodes well for these. And the eventual clinical trial is a very small and very simple trial. It's to take roughly, we've done the power analysis, if we had 25 patients with congenital X-linked NDI and simply put them on placebo or NDI-5033 for 3 to 4 weeks and then cross over. So half would start on drug, half on placebo. You then give a brief washout period and reverse it. And all we need to measure is urine output. We don't even need to draw blood. So potentially this could be, and they make lots of urine, so urine output and urine osmolality. And we reason that this could probably be done, have the patients come into a study center for some initial physical and blood work, and then they go home and they need to keep a urine diary. And actually, we can't ask them to collect all their urine. It's 20 liters. They'd be, it would flood their bathrooms. But we could do a briefer timed collection, see how much they make over 2 hours, because they make so much, and then take an aliquot and send it to a lab for urine osmolality. So assuming that turns out to be the case, you'd have to do a phase 1 trial, but the phase 2 trial could be this very short and simple study. And then after that would be done, potentially putting them open label, anyone who makes it that far open label, to just do longer term follow-up. And so that's the plan. And where we're at the moment is we submitted an SBIR in September. It'll be reviewed right before Thanksgiving, so we could have a very happy Thanksgiving or not. We're working on we've got most of the IP in place, and that will be the key for getting venture capital investment. Some of the VCs we've talked to say we're just not asking for enough money. This is like pocket change to them. We said, but okay, we can ask for more, but this is all we really need. We'll see how that goes. So this is our team. We formed a company. I mentioned Jana Klein and I are the inventors on the Emory side. Ish Khanna and Ram Pilarasetti are the medicinal chemists that we've worked with, and Rachel Hagen is our CEO. So we're the 5 founders. We are the whole company. I like to say I own 20% of nothing right now, but maybe someday I'll own 0.5% of something. And we've enlisted some advisors, Donna C., to advise us on various regulatory events, Daniel Bichet in Montreal, who has probably the biggest collection of genotyped NDI patients, Soren Riddig, who's in Denmark, who is really the European king of this disease, and we're being advised by Reef Pamuchku, who's been a very successful entrepreneur and brought in startups, so having his on our advisory board as well. So that's kind of our story. I hope it was clear enough, but if not, or even if it was, I'd be delighted to have any questions that you may want to ask. So thank you for your attention. I welcome your comments, questions, suggestions, anything.
Anybody have any questions?
So, Jeff, Dr. Sands, thanks very much. It's great. Can you hear me? Yeah, good. Thank you for a great presentation. As being a former entrepreneur and having a small company, what's your patent situation on your molecule look like? Judging by what you said that these are existing molecules. So what's your strategy to make sure that your investors are happy that you have a patent situation?
Okay. So it's an so there's 2 parts to the IP for this process. Janet and I, through Emory, have obtained a patent for the concept of using AMP kinase activators to treat nephrogenic DI. And that patent was issued in November of 17, and the company has now licensed that patent from Emory. The other side is the composition of matter patent for the NDI 5033. That patent, that molecule was developed by Ish and Ram, but when they worked for a different company. We've been in negotiations, or they've been in negotiations, to license that composition of matter patent from the company. The company that holds it really isn't interested. They want something for diabetes mellitus. They've assured us that it will happen. I think we've worked out the financial arrangements for what they would get for licensing it to us, and I think probably by Thanksgiving, if all proceeds, we will have both sides of the patent. There's another provisional patent that's been issued that is making its way through, specifically to use NDI 5033 for the treatment of nephrogenic DI, but that's still in the provisional patent stage. So in our discussions with the VCs, I think once we get the composition of matter patent licensed to the company, combined with the earlier patent that's now already been licensed, that would be enough to reassure them that we can move forward.
Anyone else have anything for Dr.?
Thanks for excellent presentation and your work. 2 questions. One is in kids, how does the homeostatic mechanism or compensation mechanism will work with the renin, angiotensin, aldosterone once they are losing enormous amount of fluids or liquids? Are they in overdrive trying to compensate for that, or are not listening to the commands for that system. And the second one is about the feedback also from antidiuretic hormone. So are they high levels screaming at the kidney to work with them because it's secondary nephrogenic in that case? So how does those two are going to be working on those in those kids?
So the vasopressin levels are most likely high. They've been measured. The vasopressin are high, but because the kidney doesn't respond, it doesn't matter how high they go, it's complete end organ insensitivity if you have complete nephrogenic DI. There are, not that we've talked about here, there are partial forms of nephrogenic DI where you may get some response, but for complete nephrogenic DI, which is the case when you have the congenital forms, vasopressin is high, but it's ineffective in doing anything. The renin-angiotensin system probably doesn't seem to be involved. It responds mostly to changes in sodium homeostasis and blood pressure. And these kids do not have a problem with their blood pressure, assuming that hydrated, then there should, and it's really the abnormality in water. Their sodium handling is normal, but as I mentioned, they have to eat a very low sodium diet. So as long as they're reasonably hydrated and following what they should, it doesn't seem that there's no mention of any abnormalities of the renin-angiotensin system. They're not known to be. There's no hypertension, for example, whether it's because they're eating low salt and they're offsetting it. But that system seems to be okay and intact and hasn't ever been reported as an issue either in the congenital form or in people on lithium.
Are specifically not working for the V2R
Right.
segment.
So the V2 receptor is expressed exclusively in the kidney collecting duct, in the thick ascending limb, and in part of the distal tubule, in the connecting tubule. And so those are the segments that are affected by the V2 receptor mutation. The reason, so let me go back, oops, maybe this was not a good idea to go backwards, but I wanted to get at your question, go back to this slide here. So that's these segments that are affected, and here, this is more or less intact, and this is all intact. And so the angiotensin II has its major effect here in the proximal tubule to reabsorb salt and water. And that's all fine, which is why I think there's really no abnormality of blood pressure or abnormality of the renin-angiotensin system. The reason thiazide diuretics are marginally beneficial is actually if you give a thiazide, you induce a mild volume depletion through action here through a sodium chloride cotransporter. If you get a mild volume depletion, then what it'll cause the proximal tubule to do, it will then stimulate a renin-angiotensin system through the volume depletion. That will increase proximal reabsorption. So normally the proximal tubule will reabsorb between 60 and 80% of the glomerular ultrafiltrate. In the presence of activated renin-angiotensin, mild volume depletion, you push it to 80%. So what does that do? It means only 20% can go to eventually become urine. The rest of it's been reabsorbed proximally. So that's the trick of the thiazide. It doesn't work terribly well, but but it does lead to some improvement in less urine output, not through changing the concentrating mechanism or any of these steps, but simply by keeping reabsorbing more of that glomerular ultrafiltrate before it can hit the loop of Henle. And that's where the renin-angiotensin is important for increasing reabsorption here. But it's isosmotic, so it's not improving the concentration. But if the net goal is to reduce urine output, it does have some beneficial effect. It's just not easy for people to eat it. And you have to eat a low sodium diet for it to work, just as actually even in essential hypertension, if you eat enough salt, you can overwhelm any blood pressure effect of the thiazide. So that's why I slid over that earlier on. It's renal fellows, internal medicine. Everybody hears me say that, and they still come away scratching their head because it's just counterintuitive. Maybe your fellows are better than ours and they understand it, but I think it's a head scratcher for everybody. But your comment really pertains to that. So thank you.
Thank you, Dr. Sands, for your work. My name is Lucas Alston for Technically Speaking, the school newspaper. After you secure the patents around this Thanksgiving, you said, when do you think that your company would have a product to human trials and then to market?
Well, you never really know. In the best of all worlds, it would be about 4 years. But that's if everything works. So I think because until you do all the pharmacology, the pharmacokinetics, the toxicology, you just never know at what point the project's going to wither and die. The other part of our model is this is therapy for life. And so you need to do longer term animal studies looking, for example, at carcinogenesis, other abnormalities. We've not seen anything in the 1 month PK or tox studies that we've done, but that's 1 month. The goal would be continue these short-term studies, get additional funding, be able to go into the longer-term studies, go to the FDA while continuing to do these longer-term animal studies. And then once the FDA gives us says go ahead and move, I think the last phase, the phase 1 and 2 clinical trial, could potentially be done in a year. So in the best of all worlds, I would say 4 to 5 years. But there's no guarantee we'll ever get that far because most things never, I'm realistic enough to know, and Russ probably knows better than I do, most things never make it. But we hope so. We have nothing so far that tells us it won't make it, but a big question, a lot of steps along the way. Yeah.
Hello. I used to do first-in-man clinical research in my earlier days, I guess you could say. And one of the things that we used to do was measure urine output for various drugs. And you were mentioning about the urine output to measure if you're seeing a reduction based on this particular disease. But I was wondering, one of the things that we did when we were measuring urine output was also to see how much the actual drug was being excreted afterwards. So I know you've done some in mice, but I didn't quite see if you've actually had any first-in-man human data.
No.
Okay. So is that some kind of a concern you guys have, or do you feel like that's not really, like, having too much, like, being that they are, I guess you can say, urinating, say, 20 liters out, is this a concern about, like, how often they'll have to maybe replenish or redose or what?
Yeah, we haven't, I think even... we haven't done any of the first-in-man studies. We also haven't, even in animals, done studies where we look at maybe once-a-day dosing is not optimal to really get at the drug half-life. Maybe it's twice a day, especially with that. So yes, we have to do all that as we move forward. So it would be wonderful if it was once a day. My gut feeling is it's going to end up being twice a day, but even in the animals, we haven't done that yet. John.
Thank you very much for that great talk. So I'm not that familiar with AMP kinase. So it would be an extension of the question asked. Is it expressed in any other tissues? Does it stimulate anything else? Is there, if you are aware of it being somewhere else in the body, what
Yeah.
potential side effect could there be from, revving it up?
So, excellent question. AMP kinase is virtually everywhere in the body. It's a ubiquitous kinase. It is, it's a heterotrimeric kinase. It has an alpha, beta, and gamma subunit, of which there are multiple isoforms of each of the three, so you can get a whole bunch of combinations. There is some tissue difference in which isoforms are expressed where. One of the things we are currently investigating is to get at the question of how do you get the specificity? I mean, if this molecule is everywhere and you're activating it presumably everywhere, when we've done the initial studies looking at tissue levels, we've done like different tissues, liver, heart, whatever, it does look like there is for some reason we're getting more activation in kidney than we are in other places. I don't know the mechanism by which that's happening. Is it that and you saw that we have 3 molecules that we looked at, one of which does well and the others didn't. So could it be that there's something about this compound that is allowing it to be more selective for the kidney than the other compounds? The AMP kinase activators for diabetes mellitus presumably are having better penetration into the pancreas or other places. So it's a question we have been asked by the VCs, why do you get specificity? And we're trying to get them some reassurance. We're trying to at least show them that there is specificity, even if we don't know the mechanism as to why. So, no, that's an excellent question that we're wrestling with.
Last question, Jeff. You mentioned 20 liters. What would be normal for a child in terms of urine output in a day?
Oh,
just to give the perspective.
Sure. It's probably a liter or 2.
Liter or 2. Great. And intake?
Well, it would match it. So a normal child, especially a baby who's either getting formula or breast milk, breast milk, you never know how much they're taking in. Of course, bottles, you're giving, babies don't concentrate terribly well, but they can concentrate enough that they're not doing this. As they get older, like certainly by the time they're able to be, well, even before they're potty trained, by the time they get over a year old, they can concentrate their urine. So it's going to depend on how much, like adult people, how much they drink. But young babies, less than a year, do not have fully developed concentrating ability. But they're getting all their nourishment, in essence, in liquid form. So they easily stay in balance and probably are making on the order of a liter or so per day.
Great, thank you. Jeff, Dr. Sands, on behalf of the college and on behalf of us here at the National Center and also our sponsors, as Alan mentioned, the SciTech Foundation and also the North Carolina Biotechnology Center, we want to present you with a small token to take back to Atlanta and to hang in your office, and I hope you'll remember us. And
Absolutely.
we really appreciate you coming here on your time and spending time with us. And actually, Gus Solis is in the back there. You've just saved him a whole 3 weeks of teaching the kidney in physiology. So thank
Glad
you
I
very
could
much
help.
for coming. Really
Thank you.
appreciate it.
Thank you all.
Just want to make a note too, and Linda and Alan, thank you very much for helping today at SciTech, that I think Jeff is 57th speaker. We're not sure, but we've got a log of all our speakers we've had over the last 10 years. Definitely we've had upwards in the high 50s for speakers. It depends. Sometimes we have multiple speakers at the same time. That's why the count might be a little off. But we have on the series this fall, following Jeff will be a fellow named Andy Hebard's coming in, who's going to talk about omegas. So that'll be November 15th at 4 o'clock here. And then on December 6th at 4 o'clock, Dr. Gary Green will be speaking. Dr. Green is the retiring president of Foresight Tech. So we'd welcome you to bring your classes, bring your students, bring everybody that you can. We look forward to seeing you. So thank you and have a good night.